In this video, we're going over the concepts for chapter 12, which is titled Amino Acids, Proteins, and Enzymes. The first thing that we are going to go over is defining what is an amino acid. An amino acid is an organic compound that contains both an amino group and a carboxylic acid group in the form of a carboxylate. As we can see in figure 12.1, that is the structure for all amino acids. All amino acids have the following components. I'm going to zoom in to look at it more closely. All amino acids contain an alpha carbon, a carboxylic acid unit that lacks a proton at the oxygen. So that's why it's called a carboxyl group or a carboxylate group. It has an amino group, and this is just a form of an amine. As you can observe, it's going to be protonated. Lastly, we have what is called the side chain. By now, we have always discussed that an R group represents the rest of the molecule. And in amino acids, that side chain is what makes the difference between each of the amino acids. We can see in figure B the skeletal structure for amino acids, where you observe the protonated amino group, the carboxylate group. Here is the alpha carbon. The hydrogen is not drawn because remember that hydrogen atoms are implied. And then you can see here a wedge representing that the R group tends to be facing us. Remember that stereochemistry is something that we learned in the previous chapter. There are 20 common amino acids. And those amino acids, depending on the different sequences, is what makes up a protein. So what that means is that the basic unit of a protein is an amino acid. We call them alpha amino acids because the carbon that we have here, in the case of the general structure that we see in figure 12.1, is a carbon with a hydrogen and everything that is surrounding it. So in general, we just call it the alpha carbon as a symbolism for focusing on that carbon atom. The side chains for the amino acids, meaning the R groups, have their own functional groups. It may contain alcohols, amines, aromatic side chains. And because of these R groups, that's what makes the individual characteristics for the amino acids. When it comes to the amino acids, we can actually represent them in Fisher projections. Fisher projections is a way that we learn how to draw sugars in the previous chapter. And as you can see, we can also utilize it for amino acids. When it comes to the amino acids, let's understand that in the Fisher projection of it, we're going to have the alpha carbon connected to the hydrogen. The top part is going to be our carboxylate group. To the left, in this case, we have the amino group and we have the R group facing the position that is pointing down. Many amino acids, I should say the majority of them, can have at least two stereoisomers. Some can have more than two. And that is because, in essence, whenever we have an amino acid, since the carbon atom, meaning the alpha carbon, is connected to one, two, three, four distinct groups, the alpha carbon in 19 out of the 20 amino acids is a chiral carbon. Now, similar to what we observed in sugars, amino acids can have L forms and D forms. As long as you have the carboxylate group pointing on the top part of the alpha carbon, the R group is pointing down in the alpha carbon, and you have horizontally the hydrogen and the amino group, then you can determine which amino acid is which amino acid is L versus which amino acid is D. So in an amino acid where the amino group is pointing towards the left, that is an L stereoisomer for an amino acid. In an amino acid where the amino group is pointing towards the right, that is the D amino acid. Understand that in nature, what predominates in terms of the D or L forms of amino acid, it is actually the L form for amino acids. So if you take amino acid supplements and you look at the bottle, you tend to see that it says L and then the amino acid that you're actually taking. Because the side chains of the amino acids have different functional groups, we can actually categorize the amino acids into four distinct groups. The categories are neutral nonpolar amino acids, neutral polar amino acids, acidic polar amino acids, and lastly, basic polar amino acids. We are actually in the next following slides going to look in detail at the side chains of the amino acid by taking table 12.1 and cutting it into the different four groups so we can closely look at the four groups or categories that you see here. Let's start by looking at neutral and nonpolar amino acids. The amino acids that are part of this group are glycine. And one thing that I should mention about glycine, which is in one of the bullet points when we were talking about the stereochemistry of amino acids, is that glycine is the only amino acid out of the 20 that does not have a chiral alpha carbon. So when I mentioned previously that 19 out of the 20 amino acids have two stereoisomers, glycine is the only one that does not. Because when we look closely at the alpha carbon, since it is a CH2 group, meaning the alpha carbon has two of the same group. That's why glycine does not have two stereoisomers. The alpha carbon in glycine is not chiral. You can see that glycine, which the three-letter code is GLY and the one-letter code is G, has a side chain of a hydrogen. Then, as we look at the other amino acids, alanine, valine, leucine, isoleucine, phenylalanine, and proline, we can actually observe that the side chains, meaning the groups that are highlighted in green, are specifically different form of hydrocarbons. So if we go back into our knowledge of organic chemistry, hydrocarbons we learned previously that are going to be non-polar because of the presence of the carbon-hydrogen bonds. So because of this, basically, these amino acids, since their side chain are alkanes or other hydrocarbons, because there's some alkanes, but we can actually see some aromatics, for example, in phenylalanine. In general, having a hydrocarbon makes the side chain and overall the amino acid non-polar. Now, there's one amino acid other than glycine that has a distinct group that we haven't observed before, and that is methionine, which is right here in the bottom right corner of the slide. Methionine have what is called a thiol ether side chain. Thiol because there's a sulfur, ether because that sulfur is in between two alkyl groups. So out of all of the side chains that we see here, we now can include that thioethers in the case of methionine is going to be part of the non-polar side chain group. The next group is the neutral polar amino acids. We can see here that the neutral polar amino acids include things that have an alcohol side chain. And we can observe that serine, threonine, and tyrosine contain OH groups. Next, we have tryptophan. And when it comes to tryptophan, we can actually observe that in a way, tryptophan, other than the benzene ring, it has a heterocyclic amine. Next, we have cysteine, and cysteine has a thiol side chain. And lastly, we have asparagine and glutamine. When we look at the functional group in the side chain, we both have in these last two amino acids amide groups in the side chain. The only difference between asparagine and glutamine is the number of carbons in the side chain. Next, we have the basic polar side chains. And from that category, we have histidine, lysine, and arginine. We can observe that histidine is going to be specifically part of this group, and we have a heterocyclic amine. Now, histidine specifically, this nitrogen atom can accept protons, and that's why this is part of the basic polar side chains. In tryptophan, our nitrogen cannot accept protons. That's why tryptophan is not considered a basic amino acid. Continuing on, when we look at lysine, we have a primary amine in its side chain. And we can see that there's an extension of carbons leading up to that primary amine nitrogen that can accept protons. And when it comes to arginine, arginine specifically is going to have a special side chain that is called a guanidinium group, which essentially what we have is a carbon that is double bonded to nitrogen. And this nitrogen is the one that can accept protons and is single bonded to a nitrogen on either side. Lastly, we have the acidic side chains. And the acidic side chains are going to be present in the amino acids aspartate and glutamate, which there are also known as aspartic acid and glutamic acid. And because these are going to be acidic polar side chains, in essence, what we have in these side chains are going to be carboxylic acids. Understand that here we just show them that they do not have the proton because in essence, since they are carboxylic acids, they have the ability to lose the hydrogen atom that used to be part of this oxygen. In other words, if we have a C double bond O, OH, what you observe here is that specifically, since the proton that can leave a carboxylic acid, let me put the R group, due to the acidity of the functional group is this one, once that hydrogen leave, this is what results as the COO. That's what we observe here.